All right, so this is the second part of the DNA structure and replication segment of this chapter. And today we're going to talk about DNA replication. DNA replication is just making a copy of the DNA. Very simple. So we're going to make an exact copy of this DNA. All right? When does a cell do DNA replication? Well, if you remember, during the cell cycle, there's three parts to it. There is the interphase, a little space here, and there's mitosis. And third of all, cytokinesis. Okay, so the parts of mitosis were prophase, metaphase, anaphase, and telophase. Okay? Do you remember the parts of interphase? There's three of them. We had G1, S, and G2. Okay? Now, I'm not going to ask you everything about the cell cycle. I'm just reviewing this here because it's important information. Interphase is the part of the cell cycle where the cell is doing its normal jobs. If a cell is going to divide, if it's going to go through mitosis, if you remember, this is division of the nucleus. This is division of the cell. Cell's actually splitting. Here we're just splitting the nucleus. Well, in order to split the nucleus, we have to copy our DNA. We have to do DNA replication. And if you recall, do you recall when that happens? I feel like I'm one of those little kid shows, you know, where is the apple? Yes, the apple's on the tree, right? Kind of what I feel like. S is what we call the synthesis phase. Synthesis. Or in other words, the replication. They're synonyms. This right here is where the cell copies its DNA. It's during interphase before the cell, because in order to split the nucleus into two nuclei, you got to copy all the stuff in the nucleus, which is the DNA, right? And then we go through the rest of the cell cycle. So there's a little bit of a review of stuff we talked about before about the cell cycle. So it happens during S phase of interphase. How does it work? Okay, so I'm going to go ahead and draw a picture of it. I'll go ahead and put it up here as well, so you'll be able to see how it works. But I think the easiest way to do this is to give you a picture of it. Let me say something about the picture I'm going to draw. It's really not 100% accurate. It's a picture that I use, that I've drawn for many years, because it simplifies the whole process. I will show you a little bit later in this video. I'm going to show you a animation of how it actually occurs. But for right now, just stick with my little cartoon version of this, and I think that it's going to help you to really learn this quite well. All right? Okay, so, sorry about that. The heater's on. I didn't want it to be too loud, so hopefully it'll turn off in a second. All right, so here we go. If you notice what's happened here, this is my DNA molecule. Okay. I'm going to go ahead and put, there's a phosphate, we're going to put a phosphate right here on the bottom. So here's all my phosphates. This is a little bit of a review for you. Hopefully you remember this. Hopefully you've done, maybe studied a little bit. Now I can't have a phosphate on the end, right? Because if I have a phosphate here, I cannot have a phosphate on the other end. And since these are kitty corn, now this DNA has been unzipped, since this is a phosphate here, that means I'm going to have a phosphate here. Kitty corner from it. Remember, I can't have a phosphate. I cannot have a phosphate right here. And if you remember, so we got a few parts here. We have a phosphate, a sugar, and then our nitrogenous base pairs. Here's a sugar and a phosphate, and so on and so forth. Okay? All the way down the line. And we've unzipped the DNA molecule here. All right. I'm going to go ahead and label my nitrogenous bases on this DNA molecule. So we're going to say if this is an A, okay, and then this is C, G, A, T, T, C, G, C, T, G, A, C, and I've just made up some random code of DNA here. The complementary nitrogenous base for A is going to be T. So this one's going to be the complementary one for C is going to be a G. C, T, A, T goes with A, C goes with G, G goes with C, C goes with G, T goes with A, G goes with C, T goes with, or A goes with T, and C goes with G. So these should all be complementary to each other. We've just unzipped them. Well, what are we trying to do? We're trying to create a brand new DNA molecule that was exactly the same code as the old DNA molecule. Okay, right here, I'm going to add a little funny looking guy right here. This right here, this blob thing that I put on there, represents what we call DNA polymerase. This is an enzyme. It is the enzyme that unzips the DNA molecule, so it's moving in this direction. It unzips the DNA molecule. It allows new nucleotides that are floating around in the cytoplasm to hydrogen bond to their partner. And then it's going to create a covalent bond between the sugar and the phosphate of those nucleotides to create a brand new DNA strand. Okay? So once DNA, notice I've already got it unzipped here. Our DNA polymerase has unzipped this DNA molecule. And now, floating around in the nucleus of the cell are tons of these nucleotides. Make sure I get it right. Okay? We're going to have G's and C's and T's and A's, and put another C here, another A here. There's tons of C's and G's and T's and A's, nucleotides, floating around in the nucleus of the cell. Where do these come from? Well, a lot of these you get from the food you eat. So when you eat anything that's made of cells, which is pretty much everything we eat, you're going to pick up these nucleotides. They go through your digestive system and then end up in being transmitted to your cells where you can recycle them and make new DNA out of them. So, as you know, what will happen is we got a C here, so what's going to bond to it? We're going to get a G. And here we've got an A, so here we're going to get a T. We got a G here, so we're going to get a C that comes in and bonds to this one. A T will always bond to an A, and a C will always bond to a G. They have those hydrogen bonds, if you recall. I'm going to make that a little smaller. So this was a G, that means it's going to be a C. This was a T, this is going to be an A. This was a C, this is going to be a G. G to a C to a G, a T to an A. So here is my new strand that's forming. Same thing's going to happen up here. We're going to get a C. Right here, we're going to get an A. Right here, we're going to get a G. T. Move this guy out of the way here. A C. A G. C. And a T. Okay. These nucleotides, these nitrogenous bases, are attracted to their partner. And they will automatically come in and line up with their partner. Well, if you notice, this code is the exact same as this, right? GTCAG, GTCAG, CAGTCG, CAGTCG. So what happens is you get an exact replica of the original strand when these new nucleotides come in and connect to their partners. Okay. Well, these nucleotides aren't fixed in place yet. They're just kind of stuck there temporarily. They could fall off at any moment. So the other thing that DNA polymerase does is it creates a covalent bond between the phosphates and the sugars, right here, of each one of the nucleotides. A little sloppy there, isn't it? And the same up here. We create a covalent bond here. Right there. A covalent bond gets produced. That's going to hold the DNA together and make it permanent. DNA polymerase is what does that. It's going to link these nucleotides together with that covalent bond. Okay? In my college class, we learned the name of that covalent bond. In this class, for regular biology, 10th grade biology, we don't care about the name of that covalent bond. We just know DNA polymerase, so it's some of its jobs. Okay? In fact, maybe I can put this up here now. The enzyme DNA polymerase is going to unzip the DNA molecule. I showed that. New nucleotides come in and they bond, they hydrogen bond with their corresponding nitrogenous base. A with T, C with G. Adenine with thymine, cytosine with guanine. DNA polymerase then runs along the strand and it connects the backbone with a covalent bond right there. Okay, this is important. The original strand, so here's the original, here's the new strand. Okay? The original strand is used as a template. You will see that word a bunch. A template is a pattern. So you use the original template strand to create the new strand. Okay? Same thing here. Here is my template strand here. And right here is the new strand. Okay? So, template strand and the new strand. That's really important. You will see that on your quiz. You'll see that on the test as well. One last thing that's really important. If you notice, the new DNA, which is identical to the original strand, okay? The new DNA strands are both half old, half new. We call this semi-conservative replication. Okay? It's the idea of a newly synthesized strand of DNA being 1/2 old strand and 1/2 new strand. Okay? Yes, you will have that on a test or quiz. Semi-conservative replication. So you really don't have to know this part. I give this part because the animation that I'm going to show you will make more sense. In my college class, so if you want to learn, you take, hey, I'm going to take college biology next year, then maybe this is something you want to pay a little close attention to. But if you're just like, I'm probably not going to take college biology right now, you may not have to pay real close attention to what I'm going to teach you. This part will not be on the test or quiz. I just give it to you just mostly for fun, right? Okay. Anyway, DNA polymerase can only ever add a nucleotide onto the 3' end, the non-phosphate end. So if you notice right here, there's no phosphate here. We know kitty corner, there can't be a phosphate. There's a phosphate here, which means kitty corner, there's a phosphate. Phosphate's here, that means there's a phosphate kitty corner. No phosphate here, that means there's phosphate kitty corner. Or non-phosphate kitty corner, sorry about that. You can't add a new nucleotide onto a phosphate end. So as DNA gets unzipped, you can only add a new nucleotide onto this bottom strand. You can't add one onto the top. So what the DNA does is it unzips a big portion of it. The bottom strand here just adds new nucleotides as it gets unzipped, and the top strand starts here and then works its way backwards to where it left off. Okay? These are called Okazaki fragments. I love saying that. Right here. An Okazaki fragment is a fragment of DNA that it unzips a whole bunch, and then it starts here and then works its way backwards to where it left off. So it's a little bit of a, like I said, it's not something you need to know, but I'm going to show you an animation in a second, and in that animation, it shows the Okazaki fragments, and then students are like, what? I don't even know what it's talking about. For high school, 10th grade biology, all you really have to know is that DNA polymerase unzips it, new nucleotides come in and hook up to their partners, and the DNA polymerase then links the backbone, the sugar phosphate backbone together of those new strands, creating brand new DNA strands that are exact copies of the original strand. There's also a leading lagging strand. I'm not going to get into it. Okay. So I'm going to show you some animations. Let me go to my computer to get this up. I'm going to go ahead, hopefully you can hear me back here. I'm going to go ahead and just do this computer model. It's pretty cool. This is just a minute. It takes about 1 minute. Let me see if I got the right one. Yes, I do. This is so cool. Make sure my volume's up. And you can hear that okay.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
Great. So that's a really simple animation, actually, compared to how it actually is. But it's pretty cool to see that you have this really complicated enzyme that's grabbing hold of things and moving things and clamping down on it. And then you've got that DNA on the bottom that's streaming out. That would be the strand that's just being added on as it gets unzipped. And then you got the other strand that you have to unzip a whole bunch, and then you got to start here and work your way back to where you left off. Those Okazaki fragments. So, okay, great. Let me go ahead and get back to my animation here, or my PowerPoint here. Okay, this is just a little review of how cells form chromosomes, because what's going to happen, one strand of this new strand of DNA that was copied becomes one sister chromatid of a chromosome. The second strand of the DNA that was copied becomes the second strand, or the second sister chromatid of a chromosome. So you can imagine if I have a chromosome right here, okay, one strand, let's say this top strand, became this chromatid, and this bottom strand became the other chromatid of the chromosome. If you recall, sister chromatids are always exact copies of each other. Well, look at my DNA. This is an exact copy of this. We're not going to worry about this clip on coiling chromosomes. Okay, that is the end of the second section of this chapter, which is over DNA replication. Again, if you have any questions, concerns, please send me an email. I'll try to get back to you just as soon as I can. You can try to call my classroom looking at those extensions that Tyler Telford sent out. But please just let me know if you have questions or concerns. There should be a worksheet on Canvas that goes along now with this, with these last two lessons I've given, the one on DNA structure and DNA replication. So there is that, and then there's going to be a quiz. I will let you know what we're going to do on those quizzes here in the next few days. Just watch the announcements that are going to be on Canvas. Thank you.